DNA replication. First part of DNA replication is the DNA opens up and separates the strands. Helicase is what separates the DNA. Each strand continuously replicates while the lagging strand moves in the opposite direction to form Okazaki fragments. And the new DNA comes in and duplicates with the old DNA. Primers are a small nucleotide for DNA to start and end. Polymerase, which is an enzyme that brings it back together.
DNA is used to make proteins. It goes through a process called transcription. First, helicase is used to unzip a section of DNA and a strand of mRNA is created by RNA polymerase. This strand goes along with the nitrogenous bases of one side of the DNA. After the mRNA is created, it goes out of the nucleus and into a ribosome. Next, it goes through a process called translation. The ribosome reads 3 letters at a time, and for every 3 bases it reads, it attaches an amino acid. The amino acid comes from the tRNA, which float around the cytoplasm. When 3 letters from the mRNA match ones from the tRNA, a tRNA attaches an amino acid to the ribosome, starting a strand of amino acids. When the ribosome is finished reading the mRNA, there are multiple amino acids attached to each other, making a polypeptide.
Endonuclease is a restriction enzyme. A restriction enzyme just is essentially a protein that basically stops or cuts DNA molecules at specific sequences. So you can see GATA is getting stopped, which stops it. So this is the solution that we made. We use soap and salt to make and then we mix the two together. That is what it should look like. And then this is just the strawberries in the solution, and then we just filtered it out. This is us then pouring ethanol in with it. And then this is what the end product, and you should be able to see the DNA. So this is extraction of DNA. So basically, that is a lipid. We went into the lipid, we used soap and salt to break the cell membrane. And then in order to actually extract the DNA from the cell membrane, we had to use ethanol to extract the DNA from the cell membrane.
A polymerase chain reaction creates multiple versions of DNA. It can be used to create exact replicas of genes manually. BP is unique to every individual. Using BP, you can tell who was at the crime scene using their DNA. If the BP matches the crime scene, you know that it was them.